Good afternoon. My name is Cynthia Moss, and Karen David invited me to co-organize this special session, Enhancing Species Diversity in Neuroscience Research. And I want to offer special thanks to Karen and to the NINDS for their support of comparative research in neuroscience. So with the advent of genetic tools, research has largely focused on a small set of animal species. And though there have been many important advances through the study of these animals, it's essential to focus on the questions that drive neuroscience research and choose the champion animal, that is, the animal that is best suited to answer a particular question. So today we'll hear about research that adopts this approach. Specifically, we'll learn about new discoveries that come from studies of diverse animal subjects. Marmoset, bat, prairie vole, Siamese fighting fish or betta fish, shellless mollusks, nudibranch, and octopus. Each of these species offers key advantages to studying particular problems, an approach articulated by Nobel laureate Alexander Krog about 100 years ago. Krog stated, "For such a large number of problems, there will be some animal of choice or a few such animals on which it can be most conveniently studied." And major discoveries in neuroscience have emerged from research that takes advantage of species specializations. And I'll just mention two that are certainly familiar to many in the audience. So by measuring changes in electrical charges in the axon, Hodgkin and Huxley discovered how nerve impulses are exchanged between cells. The work was made possible by selection of a suitable species with a giant axon, the squid. Many of you are also familiar with the elegant studies of learning and memory carried out by Kandel and his group, and this work exploits advantages of the behaviors and nervous system of the sea slug, Aplysia. So the goal of this session today is to inform the community of the Brain Initiative support for the diversity of species used in neuroscience research. By leveraging advances in genetics and neuroscience tools, new species are being explored and developed for their utility in addressing outstanding neurobiological questions. This symposium will showcase research that features non-traditional species. And specifically, you'll hear about some of the questions neuroscientists are asking and the animals which hold the answers to these questions. After the research presentations, we'll have a discussion that focuses on the challenges and opportunities in expanding the diversity of species in neuroscience research. This discussion will be moderated by Zoe Donaldson. So let me briefly introduce today's speakers. First, Corey Miller from University of California, San Diego, who will be speaking on spatial and social behaviors in marmosets. Angela Salas from the Johns Hopkins University will then talk about spatial tracking and social behavior in bats. Zoe Donaldson from the University of Colorado will present work on social bonding and parenting behaviors in prairie voles. Andreas Bendesky from Columbia University will talk about work on fighting fish and their aggression in betta fish. Paul Katz from UMass Amherst will discuss foraging sensory motor behaviors in mollusks. And finally, Galit Paled from Michigan State University will discuss grasping behavior in octopus. So now let's turn to our first speaker, Corey Miller, who will tell us about his work on marmoset social cognition.
Okay, so first I just wanted to thank Karen for organizing this symposium on expanding species diversity in neuroscience. Where really one of the primary motivations is to showcase all of the less traditional animal models in neuroscience and sort of expand on why it's really important to motivate your research questions with the appropriate model species, because every animal does all of these really interesting things. And one of the traditions has been to simply squeeze any question into into a small subset of animals, and we can really learn a lot by expanding on that. And one of the things that's important to say with all of these diverse species is I, like all of the speakers today, are very find it very important to uphold the highest standards in animal welfare in our research. All of the kind of work that we do is scrutinized at the federal, state, and local level, and is upheld very high standards for research. All of these are approved by our IACUC, which meets not just the animal welfare considerations, but also high levels of reproducibility and rigor in the research. And one of the key motivations for doing animal research for any taxa, including the ones that are discussed here, is because animal models have played such a crucial role in the development of disease treatments and interventions. And this has been the case in the past and will continue to be for the future. So the animal model that I work on are common marmosets. So this is a small New World primate. They've been around in biomedical research for quite a long time, including neuroscience, but about 5 or 6 years ago, they really started accelerating in their usage. And part of the reason for this initially was that they had a particular amenability to the modern molecular toolkit that's available with imaging and viral vectors and chemogenetic approaches. And so this sort of brought them onto the stage. But in addition to this, one of the reasons that they've expanded is that they're a small-bodied monkey, which gives certain advantages for logistical considerations, but they're also a primate. And so they have the entire shared architecture of primate brain, which has certain implications for better understanding humans. One of the things that's really emerged as a key part of why we use marmosets is that because they're small, they're amenable to a range of neural and behavioral recording preparations for freely moving animals, which allows us to look at more naturalistic behaviors. So traditionally, like you can see here, the way that people have generally studied primates is in restrained context where the animals are looking at screens and performing some kind of task. But this is not necessarily fully representative of what primates do. So if you look at the breadth of really a species' entire repertoire, most of what we've learned about the brain comes from the kinds of conditioned behavioral tasks that are common in primate research, as well in lots of other species, and the very specialized behaviors that are sort of classic neurobiological models like songbird learning and echolocation. And both of these really are benefited by the fact that animals repeat the same behavior over and over at high levels, which allows you to record neurons and look at correlations between them. But each of these sort of brackets at the end of the behavioral repertoire really don't represent even everything that an animal can do. And one of the nice things in modern neuroethology, and that you'll see in the work that we do and lots of other people, is that our capacity to be able to quantify and characterize behaviors now is such that we don't have to only rely on these very high frequency behaviors, because we can look at behaviors over long time scales, even ones that occur at low frequency. With respect to marmosets, this has been a real boon, but also simply because one of the real key features of marmosets where they really excel is in the social domain. So they have a lot of characteristics in their cognition, communication, and social behavior that are rare in other non-human primates, but have lots of parallels in humans. And this includes things like prosociality, imitation, they're highly vocal, and so forth. And part of the reason that this is important is ultimately that the primate brain is a social brain, right? Like I always like to say, like, it's the only thing that primates do well. We're slow, we can't jump, we're not very strong, our sensory systems are pretty mediocre, but we're really good at lying and cheating and forming alliances and all these things that, really we enjoy watching in television and movies. It's because that's what our brain is tuned to do. And it's not that other animals aren't social. There are social animals in the vertebrate and invertebrate domains, lots of really interesting types of behavior, but the social landscape of each species, each taxa is very different. So the types of challenges that they face, the way they model that social landscape and how they would solve those are all very different. And if you want to understand how we do this, the best case for doing that is in species that view and see the world in the same way that we do and model that social landscapes accordingly. And this is one area where marmosets have been really useful. So my work in the lab kind of brought broadly touches on 3 areas. It doesn't seem like these are all socially related, but they are in the long term. So part of this is just building up the basic blocks of how they function individually, because the long-term goal is to really see how they're integrated into the social cognitive processes of what this animal can do. So we work on 3 areas, all motivated by studies that we do at our field site in Brazil with the natural behavior of the animals, on active vision, where we leverage on the marmoset's natural hunting behavior, and their spatial navigation and social communication. So I just want to touch on a few of these and give you some examples for this. So marmosets are vociferous hunters. They are constantly eating insects. So insects account for about 50% of their diet in the natural habitat. And what you can see here is it's an animal that's reaching out and grabbing a grasshopper out of the grass. So one of the areas that we're most interested in terms of vision for this are the visual motor integration that's involved in this, and also how motion changes, right? So each of these are very different from how these things have been studied in traditional lab studies, because we're really asking how animals actively navigate through the world and what impact this has on their visual system, which is, you know, very different than looking at a static screen. We brought this behavior into the lab in collaboration with Austin, Alex Hawk. And what we've done is we want to be able to take this behavior in the lab and use these great neural network behavioral tracking models like Deep Lab Cuts here, in order to better characterize the fine details of what the body is doing and what the visual system is doing. So what we've done in my lab is build this simple arena with multiple cameras where we're training the network on all these different camera views, which increases our ability to be able to track the animal as it moves in 3-dimensional space, including all of its limbs and all of its eye positions. So this is exciting endeavor, and the next step here will be recording at different levels of the visual system. Separate line of work, which is focused more on hippocampus, relates to how different ways that primates explore and navigate the world integrate. So place cells are very common. They've been found in virtually every mammal that they've been studied in. So we recently reported that this was the case in marmosets. Like other primates, marmosets also have a mechanism for encoding information in the visual system, or encoding spatial information through visual encoding. So these are spatial view cells or visual place cells. And one of the things that we see in a lot of these basic mechanisms is that the way that primates distinguish between exploration and navigation may be somewhat different. So they seem to be exploring through vision and then navigating through by encoding their position within that cognitive map. And one of the key things that we're really interested in is how do these different streams of information integrate in primate hippocampus. So for this, we built a 3D arena. We're using different video tracking technology. So this uses about 20 different cameras. And part of the reason that it may be a little bit difficult to see here is that using this technology, we can not only measure where the animal is in space, but their gaze direction. So the red line here is tracking the animal's gaze, and we can project that into space. The next step of this is integrating a head-mounted eye tracking system, which will give us greater resolution on this. The last piece of work that we do is on social communication, and we worked with these conversational exchanges in marmosets for a number of years. They're socially learned during autogeny. They're governed by rules just like ours. And one of the things that we've done is to have an experimental probe into natural behaviors that we replace one of these monkeys with software that behaves like a monkey. So we call this a virtual marmoset. It allows us to directly engage the animals in their conversational exchanges. And this is nice because in a lot of these sort of just natural behavioral tracking, you don't have the experimental interventions that you might want. So one version of this that we're just writing up now is that we've developed a marmoset cocktail party. So here, what you have is the animal is trying to talk to one virtual marmoset that's specifically trying to engage them in a conversation, and then there are multiple other virtual monkeys that are ignoring them and engaged in their own conversational exchanges. And through doing this, what we've been able to find is that the marmosets are using very similar perceptual mechanisms as humans, as well as adapting their behavioral strategies to optimize their communicative efficacy. So just in sum, marmosets have really taken off, and one of the reasons for this is because of their amenability to these natural behavioral kinds of paradigms, and that, marmosets, because of this, have expanded what we can even study in the primate brain. And one of the key features that moving forward that will be a target for all of this research is their social behavior, cognition, and communication. And next up will be Angie Salles. Thank you.
Hi, my name is Angeles Salles, and I'm a postdoc at Dr. Moss's lab at Johns Hopkins University. And today I'm going to talk to you about bats, brains, and behavior. The first thing I want to discuss is why bats. Bats are auditory specialists that produce ultrasonic vocalizations and listen to the returning echoes to build a scene of their surroundings. This is what we call echolocation. This active sensing system allows us to record and analyze the signals that the bats produce. This means that we can have full insight into the sensory information that guides the bat's behavior. They are social animals that, depending on the species, can even roost from a couple of individuals to several thousand. And they use their voices not only to navigate, but also to communicate with each other. Furthermore, bats are mammals whose brains share many similarities with that of humans, allowing for comparative work in auditory processing. Comparative studies can help separate specializations from general principles. The bats we work with are the big brown bats, which are a North American insectivorous bat that produce sound frequencies between 14 and 120 kilohertz. For reference, human hearing can only go up to as high as 20 kilohertz, so you would only be able to hear the lowest sound frequencies produced by these bats. What I'm showing you here is a spectrogram of some echolocation calls produced by this bat. A spectrogram is a way for us to visualize the calls the bats produce. We can see the sound frequencies that occur over time, and the color represents the intensity, with the greatest energy shown in red. The foraging bat controls call parameters and call timing in response to echo information in the environment. A traditional capture sequence shown here involves a search phase with longer duration calls. Then the bat locates the target and the approach phase begins. Here, the bat shortens the duration of the calls and starts emitting them more frequently. As the bat prepares to capture the prey, they need a fast succession of short duration calls with lower sound frequencies that's called the terminal bus. I'm going to show you now a video of a capture sequence in the laboratory where you'll see the bat approach from the left of your screen and sweep to the right as it captures the prey. At the bottom of the screen, you'll be able to see the spectrograms of the calls you'll be hearing. The video has been slowed down for you to be able to hear these echolocation calls. Bats are not the only animals that must track targets. Most animals use vision as the primary sensory modality to track moving targets. This gives them a continuous stream of information. Like Corey showed you before, the marmoset uses vision to track the grasshoppers it eats. Bats, as we mentioned today, use echolocation to build a scene of their surroundings. That is, they use discrete acoustic snapshots to track moving targets. Building internal models of the trajectory can allow for the prediction of future locations of a target, and in this way, may improve the performance when tracking moving targets. In the lab, we can exploit this active sensing system to investigate how bats track a target moving across their acoustic field. This is a top view cartoon of our experimental setup, where we have trained the bats to perch on a platform and track a target that is moving from the left to the right, as you can see here. The bats emit echolocation calls and aims its head towards the moving target. I'm going to play now a video of what this looks in real life. This video also has been slowed down for you to hear the echolocation calls, and you will be seeing the target move from the right of the screen to the left and see the bat accompany the target with the echolocation calls and head movements. We have infrared cameras so you can see the bat, but the bat is actually doing this in the dark. So it's relying in echolocation to track the target. Our data shows that the bat is always aiming its head ahead of the target motion. We asked ourselves then, how is the bat able to keep ahead of the target? For this, we developed mathematical models to explore different hypotheses. we developed a non-predictive model where the bat aims its head based on the last echo return to estimate target position. As the sound takes time to move across the air and it only receives discrete snapshots, the information the bat receives can be stale, and the target may have already moved from that position. On the other hand, we develop a predictive model which is based on the bats estimating the velocity of the target based on a sequence of echo returns. The bat's tracking behavior reliably aligns with the predictive model. To test this predictive strategy used by the bats to track moving targets, we added an occluder to part of the trajectory. Here, as the target moves across the acoustic field, part of the trajectory is occluded, and the bats can't receive acoustic information to know where the target is during this portion of the trajectory. We found that bats rely on the predictive strategy to continue to track the target as it emerges from the occluder. If it was using a non-predictive strategy, the bat's head would lag behind the target. The bat's target tracking behavior in the occluded trials again reliably aligns with the predictive model. But targets moving in the environment are seldomly isolated. Usually, an auditory scene is composed of many moving auditory objects and background noise. The ability of humans and other animals to sort acoustic signals from different sources in a dynamic environment is called the cocktail party effect. As I mentioned at the beginning, bats are social animals and hunt in their own little cocktail parties, where not only they have to discriminate the echoes from their own vocalizations, but also attend to communication calls emitted by roostmates. Next, you'll see a short video of what these bat cocktail parties looks like in the wild. These bats are hunting for insects in close proximity to each other, and you will be able to hear their calls through the output of a bat detector that makes them audible for human hearing. In these bat hunting cocktail parties, bats are emitting echolocation and communication calls. As you can see here in these spectrograms, some of the social calls emitted by bats may resemble echolocation calls and overlap in acoustic features. Yet, they have very different meanings, and the bats are able to discriminate them. Thinking about it in humans, a situation I get all the time when presenting my work at conferences is that when people ask me what animal I work with, I say, bat, and immediately they look at me quizzically and say, rat? Like the bat's calls I showed you before, these two words are similar but have different meanings. And if you add to the mix a noisy environment, the discrimination is even harder. We recorded neural activity from the bat's midbrain and found neurons in the bat brain that respond differentially to similar vocalizations that have different behavioral relevance. Some neurons are selected for social calls, and some are selected for echolocation calls, regardless of their acoustic similarities. This differential selectivity contributes to the discrimination of calls that share acoustic features but differ in meaning. Working with bats can give us insights into how different animals track targets, furthering the understanding of predator-prey interactions, and it can advance the understanding of how the mammalian auditory system works to track auditory motion and discriminate sounds in a dynamic world. This is also relevant for humans, especially those who rely on hearing for navigating their surroundings, or whose hearing is impaired, and new technologies in auditory processing can improve their way of life. Our research program focuses on investigating natural behaviors, which in turn allows the bats to exercise and perform natural tasks such as flying and prey capturing. With that, I want to thank the lab, especially my PI, Dr. Cynthia Moss, the NSF, and our other funding sources. And now we'll hear Dr. Zoe Donaldson speak about social behavior in the prairie voles. Thank you.
Thank you so much for the opportunity to be here today. I'm going to talk to you about my lab's work, which is focused on understanding the neural basis of adult attachment or pair bonding by studying a rodent species. I'd like to begin by thanking the people who actually carried out this work, as well as the funding sources that made it possible. And consistent with this panel, I want to start with the question of why voles. And within the context of my lab, the answer to this question is that voles exhibit behavioral traits that are not found in other more commonly studied species, such as mice. However, they are a laboratory amenable species, and they're evolutionarily close enough to mice that many of the technologies that have been developed to investigate brain function transfer well to this species. Ultimately, I'm interested in understanding the neuronal basis of pair bonding, or what in humans we might refer to as falling in love. However, there are some limitations to trying to study the cellular basis of this behavior in humans, as we can't yet put tiny microscopes into human brains. And so one of the questions that I get a lot is, why don't I study mice? And the simple answer to this is that mice don't form pair bonds amongst mating partnerships. And you can't study something that doesn't exist. And so instead, my laboratory espouses Crow's principle, which states that for such a large number of problems, there will be some animal of choice or a few such animals on which it can be most conveniently studied. And today I'll argue that prairie voles represent a species on which we can most parsimoniously study adult attachment. So in the wild, a male and a female prairie vole will meet, they'll mate, they'll establish a territory, share a burrow, and both parents will help take care of the offspring. This is in contrast to the majority of mammalian species, where the female will go off and raise the offspring on her own. And as such, these represent an ideal species for studying the biology that underlies social monogamy. But how do we take something complex like social monogamy, which is ultimately a social organization system, and turn it into something that we can quantify within the laboratory setting? My lab and others take advantage of something called the partner preference test, which ultimately quantifies an affiliative preference for a particular individual. And so what you can see in this video is that the test animal is placed in the middle of this 3-chambered apparatus. It can then freely explore, and the first interaction that you're going to see is between this test animal and a novel opposite-sex volus never encountered before. And here you can see that this is a somewhat aggressive encounter, and that's characteristic of prairie voles who formed a bond. And they're typically only affiliative with their partner. And so here you can see this sort of more positive social interaction. They'll spend the majority of the time huddling with their tethered partner. This test typically lasts for 3 hours, and we can quantify the amount of time that they spend huddled with their partner. If they formed a bond, they'll spend substantially more time with their partner than with the stranger. If they haven't formed a bond, or if this test was done with a species that's not capable of forming bonds, you would see that they don't distinguish between these two individuals. So if you think about what this test is showing us, ultimately, it's that bonds are cemented over time by the desire to reunite with an absent partner. And so with this premise, one of the first questions I asked when I started my laboratory was whether or not patterns of neuronal activity in the nucleus accumbens reflect partner reunion in pair-bonded voles. And we focused on the nucleus accumbens because of its classic role in motivation and reward, and because it's a part of the brain where neuromodulatory signaling is required in order to form a pair bond within this species. In order to undertake this work, we had to pioneer the use of in vivo calcium imaging within this species. And in order to do that, we actually use a viral vector in order to transduce the neurons such that they glow in the presence of a calcium signaling event. We can then use a tiny microscope mounted on the animal's head to detect these events. And so this video here is actually showing you what it looks like when you look through the microscope. The sort of spider webs are individual blood vessels within the brain, and the occasional flashes of light that you see are individual neurons exhibiting calcium signaling events. The advantage of this approach is that it enables us to be able to monitor neuronal activity in freely behaving animals, such as prairie voles that are performing a partner preference test. And so this allows us to draw on a rich literature regarding the biology of partner preference using a partner preference test. So we undertook a longitudinal study in which animals performed a partner preference test, initially at day 0, where they were choosing to interact with one of 2 unknown individuals. They were then paired with one of those individuals, mated, and then cohabitated with that individual for the remainder of the study. We then carried out an identical imaging session at a short-term time point post-mating, 3 days after mating, and at a long-term time point 14 days after that. Each individual time point looks like this, where the behavior is shown on the left and the neuronal activity is shown on the right. And so the test animal here is seen as interacting with the novel. It goes over and starts interacting with its partner. And on the right, what you can see is the video has just been enhanced to make it easier to see these calcium signaling events. One of the first things we learned by looking at the longitudinal within animal data here is that prairie voles, like humans, exhibit sort of an increase in the strength of the pair bond given time. And so here they don't distinguish between the partner or novel individuals, which are both unknown individuals at the naive time point, but after pairing and mating, they show a preference to spend more time with the partner than with the novel individual, and this preference becomes stronger over the course of time. So we wanted to ask the question of whether or not we could identify cells whose activity might underlie this desire to be with the partner. And in particular, we focused on cells that might signal the desire to approach a partner that you're not currently in contact with. So we identified individual neurons, we identified calcium signaling events within those neurons, and then we simply asked the question of what does the animal do after the calcium signaling event. And we were particularly interested in neurons that may signal partner approach, as shown here, where there's a calcium signaling event, and then the test animal gets closer to the tethered animal. Now, ultimately, the assignment of a neuron to approach neuron or departure neuron is not based on a single calcium signaling event, but rather the aggregate across the test session. And so that's shown here, where we can map the calcium signaling event onto the blue circle. And then I've shown with the arrows the trajectory of the animal after the calcium signaling event, such that for the approach neuron, the majority of the time, calcium signaling events are followed by an approach towards the tethered animal. For departure neurons, it's away from the tethered animal. And then for neutral neurons, there's no directional selectivity. Using this approach, we can identify approach neurons and departure neurons independently for the partner tethered at one side of the apparatus and the novel animal tethered at the other side of the apparatus. We then asked the question of whether or not there were any changes within these approach cells as a function of bond formation and maturation. And what we found is that upon bond formation and maturation, there's actually an increase in the proportion of the population that's signaling partner approach and a corresponding decrease in the proportion of the population that's signaling novel approach. And this really nicely mirrors what we see at the behavioral level, whereas there's an increase in partner preference and time spent with the partner, there's also an increase in the size of the partner approach ensemble. Now, this is not only true across the multiple animals that we looked at, but we can also correlate that with individual differences in bond strength. So shown on the y-axis here is an indication of bond strength, and this correlates nicely with differences in the relative size of the approach ensemble for the partner and for the novel individual. So what this means is that we are able to identify partner approach ensembles that expand after mating, mirroring the emergence and maturation of partner preference, that pair bond strength correlates with partner approach ensemble size. And then in work that I didn't go into today, we also know that the approach ensembles are actually specific for the partner and for the novel vole, and not just approach signaling cells more generally. And finally, we know that the majority of these approach neurons have signaling events that precede approach rather than occurring during ongoing approach, suggesting that they may be involved in the decision to approach the partner or novel individual. So if we sort of zoom out, then I think what we've identified is a potential neuronal substrate located within the nucleus accumbens that may represent a physical substrate underlying partner-directed motivation. And my lab is currently using a number of other tools that have been made possible through the BRAIN Initiative in order to functionally test this hypothesis. And finally, I just want to point out that identifying the neural basis of selective social motivation has implications for a wide range of neurodevelopmental and psychiatric disorders that are characterized by either over or under attachment. And within this category of over-attachment includes pathological bereavement or complicated grief, which is gaining more attention as we face sort of the impacts of this global pandemic. So thank you for your time. Up next is Andres Medeski, who's going to talk to us about aggression using Siamese fighting fish.
I want to thank the organizers for the invitation to speak in this symposium, focusing on fascinating animals that nobody else wants to work on. In contrast to the endearing pair bonding behavior of voles that Zoe just talked about, I will now tell you about animal fighting. And to start, I will let David Attenborough tell you why we're using Siamese fighting fish.
Siamese fighting fish, or to give them their scientific name, beta splendens. And they come in a great variety of ravishing colors. So it's not difficult to understand why they are very popular aquarium fish. I used to keep them myself. They swim with all the grace of a virtuoso ballet dancer, but don't be deceived. Watch what happens when I lift this and they see one another. Because the males are so aggressive, it's illegal to put them together in the same tank. Even so, each is determined to defend its territory. They distend their fins and push out their gill covers so that they appear larger and more imposing. If these two males were kept together in the same tank, they would soon turn on each other, and one of them would almost certainly be killed.
People in Thailand have been breeding betta fish for fighting for the last 300 years. People get together to fight their fish, and the winner of a fight goes on to breed the next generation, while the loser and its family are killed. Thus, there has been very strong selection for aggressive behavior and fighting ability. Because of this strong selection for hundreds of generations, betta fish have become exquisitely aggressive. Moreover, as you saw in the Attenborough video, visual stimuli are enough to trigger and guide betta aggression, providing a unique opportunity to study the neurobiology of visually driven aggression. Today, I'll tell you about the work of two superb scientists in my lab, Claire Everett and Amy Norovich. Claire and Amy have set up sophisticated behavioral experiments and clever analysis to understand the visual features that control aggression and the underlying neuronal circuitry. To start, they developed this simple but powerful assay. Two fish are happily swimming in their own tanks, visually isolated from each other by an opaque electrochromic screen. We then pass current through the screen, which turns it transparent, as you saw here, allowing fish to see each other. Here, you can see how within seconds, fish start orienting to the fish on the other tank and mount an aggressive display, flaring their fins and opening their operculum. We video record fish from the side and from the top and use machine learning to track multiple points in the body, allowing us to quantify their behavior and their interactions. Using these tracked body parts, we then compare the behavior of fish before and after the barrier becomes transparent. We find that fish almost never open their operculum before they see another fish, but do so a large fraction of the time when they see each other. Also, fish spend most of their time right next to the wall of the tank opposed to the other fish and reorient their heads towards the other tank. We then use these tracked body parts to understand the dynamic nature of the fish interactions. For example, what is one fish doing that induces an aggressive reaction by the other fish? Here, you can see three of the possible configurations that fish adopt during these pairwise interactions, and we start by quantifying if one of these is more common than the other. To quantify these postural configurations, we calculate the angle of one fish relative to the tip of the head of the other fish. Here, on each axis, you see the orientation of each of the fish. Interestingly, the fish don't adopt random postures relative to each other. Rather, there are three main configurations the fish adopt: the red facing the yellow fish, the yellow facing the red fish, and both facing each other. More rarely, fish swim parallel to each other, and essentially never do they swim away from each other. Now, we want to understand what fish are doing when they open their operculum, a classic feature of aggressive display. We find that fish open the operculum exclusively when facing their opponent, not when being faced at. Now, we want to know what fish 2 is doing when fish 1 opens its operculum. We find that fish erect their operculum much more when the other fish is in a lateral position than when it is facing it directly, suggesting that this lateral configuration is more potent at inducing aggression. And now, these behavioral analyses of interacting fish are useful because they allow us to measure what animals do in real interactions. However, there is a limit to what we can understand because we cannot control the interactions. Fish dynamically affect the behavior of each other. To gain more control over what fish see, Claire tracked the behavior of a fish interacting aggressively with another fish and used the software that Pixar uses for their movies to create an animated movie that precisely mirrors the real fish. Now, we can play the same movie to all of our fish, providing us with a reproducible stimulus and one that we can carefully modify to study how different visual features stimulate aggression. Here, you can see fish pay close attention to the screen, almost like in a clockwork orange. For example, to measure the contribution of shape and the statistics of movement, we can compare the aggressive behavior elicited by a movie of a naturalistic fish to that of the exact same fish, but now moving in an unnatural up and down trajectory. We find that indeed, the way fish move is an important inducer of aggression, since the fish moving up and down elicit much less aggression. Now, because we play the same 30-second movie in a loop 40 times, we can determine if specific parts of the movie elicit more aggression from the observer than others. On the top, you can see a raster of every time the observer fish opens its operculum, and in the middle is a histogram of those responses. You can appreciate that some parts of the movie consistently evoke more aggression than others. Interestingly, these parts roughly correspond to when the animated fish is in a lateral position, which is consistent with what we observe in the interactions between the two real fish. Remarkably, independent fish are fairly consistent as to what parts of the movie elicit most of their aggression, indicating that these visual features generalize across animals. So now that we have a basic understanding of the psychophysics of how visual cues evoke aggression, we want to understand the underlying neuronal circuitry. To start, Amy is tracing the neuronal circuits from the retina all the way to the muscle that erects the operculum, since this muscle is almost exclusively used during aggressive display. So to do that, she injected a tracer into the eye and found only two parts of the brain that receive direct retinal input: the nucleus rostrolaterales, which is homologous to the lateral geniculate nucleus, and the optic tectum, both in the contralateral side to the injected eye. Then she injected tracer into the opercular muscle and found the motor neurons projecting from the trigeminal motor nucleus. So now that we have found the first and the last relays in this circuit, Amy is now identifying the nodes in between. And we know that some of these nodes are important because we have performed a brain-wide screen for neurons that are active while fish show aggression, and found that the dorsomedial telencephalic area, which is homologous to the mammalian amygdala, has 4 times more active cells when animals are displaying aggression than when they're observing an empty tank. So our current work focuses on developing techniques to measure neuronal computations of the dorsomedial telencephalon while fish behave in front of controlled visual stimuli that induce aggression. In this video, you can see a fish swimming with a floating head mount. We will next replace this head mount with a floating miniature microscope to optically measure neuronal activity while fish are fighting. And with that, I want to thank the whole lab, and especially Claire and Amy, whose work I share with you today. And now we will hear from Paul Katz.
Hi, it's a pleasure to be here at this meeting, and thank you for letting me speak about the nudibranch Bergia stephaniae. This is a project that I'm going to tell you about. It's a collaboration with 5 universities, and it's funded by a U01 mechanism from the Bergia Initiative. Giant brains evolved twice on this planet, once in mollusks and another time in vertebrates. There are many features that have been conserved between mammals and mollusks that are listed here. And in fact, many of these features have been discovered in mollusks. So for example, the action potential mechanisms were discovered by Hodgkin-Huxley working on squid giant axon. And many neurotransmitters were discovered in gastropod neurons. However, many features also evolved convergently between mammals and mollusks. So central pattern generator circuits, which are responsible for controlling rhythmic locomotion, which I've worked on for the last couple of decades, evolved independently several times. Muscular hydrostats in our tongues and an octopus arm is another example you'll hear about in the next talk, also evolved independently, as did complex eyes, olfaction, learning memory circuits, and social behaviors. Now, mollusks also have features that are not found in mammals, and that makes them worth studying as well. Autonomous arms is an example of that. So I'd like to compare Bergia, which is the species of nudibranch that we work on, with the octopus. Octopus have 170 million neurons in their brains. This is more neurons than a mouse has in its brain. Bergia, on the other hand, has only 5,000 neurons. So it's its little brain cousin. This is what the face of the Bergia looks like. And if we take a light sheet image through the animal using a clarity cleared animal and light sheet microscopy, this is a picture taken by Des Ramirez, who's a postdoc in my lab. You can see the cerebral ganglion, the pedal ganglion, which is where the motor command center is, the rhinophores, which are the olfactory organ, and at the base of the rhinophores are the rhinophore ganglia. If we take a cross section between through the connective between the rhinophore ganglion and the cerebral ganglion, we get this image. And if we blow that up, this is a collaboration with Jeff Lichtman's lab at Harvard, and this image was taken by a postdoc in my lab, Brandon Drescher. You see a lot of axon profiles in this connective. If we blow up this section down here on the right, you can see that many of these neurons are tiny. These are axon cross sections. Notice the scale bar is 1 micron. Many of these axons are less than 50 nanometers in diameter. Some of them are quite large, and those are small. We've segmented out this image, and we found that there are over 30,000 axons in this piece of tissue that is only 50 by 70 microns in diameter. That means that although the brain is 5,000 neurons, it's receiving over 30,000 inputs from each rhinophore. And then there are going to be tens or hundreds of thousands of other inputs to this very small brain. In some way, that's like the octopus, because although the octopus has 170 million neurons in its brain, it has 330 million neurons in its arms. Benny Hotchner at Hebrew University in Jerusalem has referred to the octopus as having embodied intelligence. That is, it's not just using its brain to make decisions and to make computations, but rather these properties are distributed across its whole body. Bergia also has this property of embodied intelligence. This is a isolated piece of skin with the serrata. These are these extrusions from the surface. They, without the central nervous system, can make directed movements towards the forceps. Now, octopus also have these very complex eyes. In fact, this part of the brain is the optic lobe. It's a massive part of the brain, and these are axons running from the eyes. In contrast, Bergia has this little tiny eye, and the eye itself is located right on the brain. Brandon has done EM images of the eye, and he's found that you can see the pigment cup. This is the lens, and then there's only 5 photoreceptors in the eye. This region down here is one of the rhabdomeric structures that seems to be the light sensing organ. So this animal only has 5 photoreceptors in each eye, yet it can use its eyes to perform visual navigation. If you put a stripe on the outside of the container, the Bergia will reliably locomote towards that stripe. And this is work of a grad student in my lab, Phoenix Quinlan. Phoenix also showed that Bergia can perform olfactory navigation. In a T maze, they will go towards their prey item, which is a sea anemone. Here's a picture of that sea anemone. My other student, Kate Otter, has shown that Bergia have to make these approach avoidance decisions when approaching its prey, because the sea anemone contains stinging nematocysts that are toxic, and they in these tentacles and also in these oconia. Bergia eats this anemone because it sequesters the nematocysts and puts them out in the serrata as its own chemical defense. One way that it can avoid getting stung is to act as a group. And so the Bergia are often seen mobbing an anemone. I'm going to skip towards later in this where they totally consume this anemone. So by working together, they seem to be able to overcome this giant predator and act as a prey, have it be their prey. Now we've seen, I don't know if that's due to social behavior of the animals or if they're smelling blood in the water. That's something that we're continuing to work on. Nonetheless, we just saw a talk by Zoe Donaldson on social behavior in mammals and the role that oxytocin and vasopressin play. This 9 amino acid peptide is very highly conserved across the animal kingdom, including gastropods and nudibranchs, where it's called conopressin. My postdoc, Cheyenne Tate, has shown that in Bergia, you can use an antibody that was raised against the vertebrate version of vasopressin and stain specific neurons in the brain. So there's a giant neuron in each pedal ganglion, and then there's a cluster of neurons in the cerebral pleural ganglion. We're currently looking at the behaviors, the social behaviors, the reproductive behaviors, and the physiological responses that are controlled by conapressin. Now, it's no secret that we've known this for decades, that many individual neurons can be identified from animal to animal within a species based on the neurotransmitter and the soma position. So this is a common feature of gastropods in general, that they have individually identifiable neurons. This has been used to study neural circuits in nudibranchs. This is the nudibranch Tritonia diamedia, and it was originally started to be studied in the 1960s by Dennis Willows. His work and Peter Gettings and Bill Frost and myself have worked out the circuit diagram going from sensory neurons to a central pattern generator to the efferent neurons that control the muscles, and this controls the swimming behavior of this animal. So that these intracellular microelectrode recordings are showing the dorsal and ventral swim interneurons that basically are producing the pattern of activity that cause this behavior. We can look at these identified neurons in several species. So the same factors, the same anatomy and neurotransmitter content can be used to identify a neuron in different animals within a species, can also be used to identify the same neuron in different species. So these animals have the same neurons, but they do different behaviors sometimes. So Tritonia swims with dorsal ventral flexions, Dendronotus and Melody swim with left-right flexions. The neurons are there, but they're wired up differently and then they have different functions. So we've been studying this for a good 25 years in my lab. And the limitation is that we're going single cell by single cell with microelectrodes. In order to overcome this obstacle, we needed a species where we can do high throughput types of behaviors. And in particular, we needed one that we could raise in the lab. And that's where Bergia comes in. So Bergia lays eggs in the lab, and it takes 2 months to go from egg to adult and back again. Our colleague on the Brain Award, Dee Lyons at UCSD, is using this developmental system to be able to inject specific precursor cells, the neuroblasts, in the embryo, and have look at lineages of cells in the adult animal, as well as be able to then, in the future, express things like channel rhodopsin or GCAMP in neurons, so that we can then have genetically encoded sensors. Another PI on the grant, Bill Frost from Rosalind Franklin University, is using voltage-sensitive dyes to record from many neurons at the same time. So this is the brain of Bergia. This is a photodiode array arranged over the top of the ganglion, and this is 20-minute recordings from over 50 neurons that we were able to do. So Bill is going to look at the activity of neurons during the production of behavior. So in summary, I've shown you that Bergia can be useful for understanding the other giant brain on the planet, that Bergia has amenable features for studies such as a simpler brain, individually identifiable neurons, and it's easily raised in the lab, and that the Bergia Brain Project is using high throughput approach to understand the neural basis of complex behaviors. So I want to thank my collaborators and the other members of my lab, and the next talk is by Galit Peled.
Okay. Thank you, Dr. Katz, for a fantastic mollusk presentation. This is the last species that we will discuss today, and perhaps the most extraordinary of them all, the octopus. I am Galit Peled at Michigan State University, and my interest in octopus started when I was an undergrad at the Hebrew University in Jerusalem. The first time I entered the octopus lab there, I saw that the octopus swam happily towards one of the students, but looking quite unpleased when other students entered the room. And that left quite an impression on me. The octopus nervous system and behavior remains mainly unexplored. But given all of its unique features, understanding how the octopus nervous system function is bound to provide information and new perspective on brain function and fuel the development of new and innovative technologies. Recently, we have received funding from the Brain Initiative to study sensory motor functions in octopuses. The octopus is amazing in so many ways. It has 3 hearts. It has blue blood. It camouflages. It has, and it regenerates almost every part of its body. It has no bones, and therefore it could move potentially in an infinite number of ways, but yet it's performing graceful and coordinated movement. Octopus existed even before dinosaurs. The first fossil to be discovered is from 296 million years ago. So the octopus has a large brain, over 500 million neurons in the arms and the brain together. It has 8 axial cords that act, each one of them like the vertebrate spinal cord. And here you can see high resolution MRI that we did, and you can see each axial cord in each of the arms. It has hundreds of suckers, each behave independently like a finger. And a disembodied arm exhibit movements that are almost identical to movement in an intact octopus. And this suggests that the octopus has simplified neural programs embedded in each arm, and it is adaptable to various degrees of input from visual and motor areas. Understanding decision-making and goal-oriented movement is a major goal for neuroscientists. These are the results, of course, of complex integration of brain and peripheral circuits. With its distributed control of the nervous system, you can appreciate that the octopus provides an unparalleled preparation to study complex and flexible motor behavior. Grasping, which is the most desirable movements that patient that lost a limb or had a spinal cord injury would like to have, and developing new brain-machine interface and neuroprosthesis have allowed adaptable and flexible gripping movement in patients is still a challenge. We are taking a holistic, multimodal approach to identify the neuronal circuits that leads to adaptable grasping behavior. This could be the foundation of developing prostheses that are adaptable like octopus arms. We are asking, what are the neuronal circuits that leads to adaptable grasping movement? Can we describe them in algorithms? And can we reproduce movement by feeding these algorithms into soft robotic prostheses? We are doing both acute and chronic electrophysiology recording. Here you can see some preliminary data where we record potentials from octopus arm. Dr. Chestek is now fabricating miniature wireless recording devices that will enable us to record multi-unit activity in an awake behaving octopus. So we will be able to identify neuronal patterns that are associated with tactile sensory stimulations, and also neuronal patterns associated with central stimulation and motor stimulation. Of course, the recording technologies that is being engineered in this project will greatly impact how can we understand neural circuits in other aquatic animals beyond the octopus, and probably in other species as well. Here you can see how one way that we could measure motion capture with very high resolution. So using 9 reflective markers spaced equally on an octopus arm and detected by 4 high-resolution cameras, this permits the capture and the analysis of movements of 3 dimensions. We are able to look at bend location, bend magnitude, velocity, strain, axial rotation in the arm, and curvature. Together, we will be able to build a model that represents the grasping behavior. Another approach in our lab is using artificial intelligence to detect motion. This will allow automated and high throughput motion detection. And we're using deep lab cut, and we trained algorithm to detect arm movement across days and weeks of recordings. We are thinking how we can take tools that were developed in vertebrates and use them in octopuses. Making an octopus that expresses GCAMP and genetically encoded voltage sensitive indicators will allow scientists to visualize neuronal activity in an unprecedented way. we have started working on producing octopus neural cultures, which will be instrumental to establish these technologies. Here you see this beautiful 3D autofluorescence imaging of octopus that we worked with imaging system. These are 25 micron section in 4 fluorescent channels. It took 19 hours to image the entire octopus. So you can see its unique structure, the big brain, even the 3 hearts. And finally, taking care of the octopus is not your average pet, and it's pretty high maintenance. It lives in salt water and it's very sensitive to its environment. So we bring live rocks that come from the Pacific Ocean, which is our octopus original habitat. These rocks have the type of algae that make the bacteria that our octopus requires. And it takes about 3 months to mature the water in the tank to have the right conditions for the octopus. On top of it, the octopus are cannibals and get very stressed out when they see another octopus. So each of the octopuses is housed individually in a 50-gallon tank. The octopus eat only live food and enjoy a diverse diet. And in fact, each of our octopus has its own dietary preferences. We grow different type of crabs and clams and shrimp. And these species eat algae. So now we have to maintain an entire ecosystem in the lab. And what you can see here is an octopus enjoying a snack. And quite happy. So at the end, I would like to thank the students, all the students that I presented the work. And I appreciate NIH for supporting my lab research that takes advantage of the unique strength of the diverse species. In this session, we showcase the value of non-traditional species for neuroscience studies at the level of circuits and behavior. We presented 6 different species with unique features and unique strength. We should continue to include other species for comparative purposes. And the BRAIN Initiative should support and facilitate experiments in these and other specialized animals, broadening the reach and scope of questions that we can ask about the brain. And finally, thank you for Dr. Karen David from NIH, Dr. Cynthia Moss from Johns Hopkins, who organized this wonderful session. Thank you for the speakers, Andres Bendeski, Zoe Donaldson, who moderates this session, Paul Katz, Corey Miller, Angie Sells, and myself, Galit Peled. The BRAIN PI meeting organizer, the NANDS Communication Office, Infinity Conferences, and Labroots. And now we'll have a session for Q&A. Thank you.
Okay, thank you so much to our presenters and also to Karen David and Cynthia Moss for organizing this. We now have a session of sort of question and answer. And so I thought I would begin with some of the bigger questions of just sort of why even bother using some of these animals. And to begin with, sort of pointing out that many of the core principles of brain function that have resulted in Nobel Prizes in the past, as elucidated by Cindy, range from sort of action potential generation in the squid to LTP and aplesia. And so we've learned in organisms, these were learned in organisms that would now be deemed non-traditional. And so this first question goes out to Paul and Galit. What are your thoughts on why a formerly more diverse set of organisms have been largely abandoned in modern neuroscience, despite the obvious successes of the past? What implications might this have for future discoveries?
I'm having some problems here because I'm getting the feedback. I don't know how to stop that. Okay.
All right, why don't we give you a moment? Galit, do you want to give this a go?
Let me see if.
Sorry. Is Paul there or?
Paul is here, but he's
keep
having some
feedback.
issues. So we're going to go ahead and we'll let you go first.
Paul? Myself? Okay. So I think that by so many great discovery has been we saw from the beginning with Cynthia's presentation. Many great discoveries have been done, of course, with the aquatic animals. And over the past decades, we really honed only on a handful of animals. And that probably, we are really missing out on the diversity of solutions that biology and evolution is providing us. So probably the Brain Initiative can make a huge difference by encouraging work on non-traditional species, for example, by specific RFAs, and it's likely, we're likely to really, and this is likely to really facilitate understanding of brain mechanisms, how other animals came with very creative solutions, and of course, facilitate the development of new technologies. And is Paul there now?
Yes, I'm here now. I figured out what my stupid problem was. So I think that the reason that a lot of the work is concentrated onto a small number of animals, and Khalid, I hope I didn't miss your answer on this, is that the tools having the communities was really important in developing the tools. But now I think that the tools have gotten democratized. And I think that we miss important concepts if we don't look broadly in the animal kingdom. Not everything is going to be like a mouse, a zebrafish, a fruit fly. And excuse me if I missed any of the other C. elegans. So it's really important to look widely and to see what the general questions are. What is nature found over and over again in evolution, and looking at convergence is really important, not just homology.
Great, thank you, Paul. So there are reasons why a few species have traditionally been very intensively studied. And I think part of that goes back to what we call Quinn's law. And so I'm going to ask sort of Angie and Corey to answer this next question. But Quinn's law states that the convenience of genetics multiplied by the quality of the behavioral repertoire is a constant for all animals. In other words, if you have animals with sort of a very charismatic behavioral repertoire, it's likely that their genetics are going to be more difficult to access. And so does this principle, do you think it still stands, or have we started to identify charismatic behavioral species that sort of break this rule?
Corey, do you want to answer first?
Sure. So you're asking if essentially we're finding more novel behaviors out in the world that warrant further study at a genetic neuro level. Is that kind of the gist of the question?
Specifically, genetically tractable, or tractable in whatever sense you want to study it.
Right. I mean, I'm probably slightly biased, right? Like I see the non-genetically tracked animals as being far more interesting, right? I mean, you know, mice are the mice and labs. I mean, there are all sorts of really cool rodents out there, right? They're like 40% of mammals and, you know, thousands of species, right? So, I mean, I think there are remarkable behaviors that are going on all over the place. You know, I'd far rather read papers on these sort of weird animals everywhere, because they do these really intriguing kinds of things that are just unexpected, right? I mean, like I think of, you know, my colleagues that study frogs. Every time that I write a paper with them and I say something like, oh, well, no one does this. And they're like, well, actually, there's this one species found in the southern parts of Madagascar that, you know, I don't know, do anything that any organism has done, right? So, I mean, in many ways, mammals have a pretty limited repertoire of things that they do. I mean, they do a few things pretty well, but, you know, there's just such a range of really interesting things out there that we know very little about.
So I think one of the traditional arguments has been that these species that do really cool behaviors are difficult or intractable from an experimental standpoint. They either can't be brought easily into a laboratory, or they're not easily modified from a genetic perspective. Do you think we're starting to identify sort of an excellent in-between in certain species where there is genetic tractability, but also these sort of very interesting and charismatic behaviors?
Yeah, I mean, I think from my point, right, so the behaviors are really the motivating force. So whether or not you can turn off every interneuron in the brain, if the behavior isn't really rich and interesting, it doesn't matter, right? I mean, especially because of the off-target effects and what the questions really are. So yeah, I think, you know, the viral tools are definitely going to be more widespread just by pure you know, capacity and money involved in having thousands of lines of transgenic animals. And so I think as those viral tools are expanding, it does allow us to dip into these species that maybe we don't have the capacity with everything that's out there in mice and zebrafish and so forth.
Yeah, I just wanted to add that, so Kari, you said like all mammals behave very similarly, and as somebody who works with a mammal that clearly has a very, very specialized behavior, I would have to argue that. But I do think that like tools like CRISPR will allow us to really like dig into this specialized behaviors and really find out like the commonalities and differences between all these mammals and all these different behaviors that they might be exhibiting.
Right. I didn't mean to say bad, you know, that was overstated for their good at everything. I mean, I think, you know, when I teach my animal behavior class and you look at all these non-mammals, you're always like, oh my God, like there are all these things that animals do that are so amazing. And then, you know, it's not that mammals don't do really incredible things. It's just, sorry. Yeah.
All right, so there's a follow-up question from one of our trainees who's been watching the symposium. And so what I'm going to do is I'm going to actually call on Andres at sort of one extreme of a species that was probably relatively easy to move into the laboratory, and Galit on the other end of probably very, very difficult. And then anyone else who wants to chime in after they've answered. But the question is, as a trainee working with one of these non-traditional organisms, how do you go about building background research and informing your work, since there hasn't been much work on some of these species? How do you balance the fact that other species might have information about key differences, and how do you convince grant reviewers to care about your species? All right, so Andres, you want to start?
Sure. I think I would agree with Corey if the behavior, at least for me, what's most important, at least initially, is that the behavior it's so interesting that then the rest becomes secondary. Obviously, if you cannot do any experiments in lab, then it's intractable, but we don't need to strive to manipulate every single dendrite of every single neuron. But if we have something very interesting to study, and if nobody else has studied it before, we can also go for the low-hanging fruit. So in a sense, it actually becomes easier if you actually get your study experimental system in the lab and you can do things, then you can discover even the most essential stuff, because not everybody's working on the same animal. And for some of these species, there is some background. There's not like there's zero, at least with Siamese fighting fish. There have been studies from the 1930s on some of their behavior, just observing them, putting them in front of mirrors. But now we can bring them into lab and use modern technologies to study them in much more careful detail. So in many cases, there is some background that one can use, or from related species, from other fish, or from other mammals, from what we study. So we're usually not working in a vacuum. Maybe there isn't something specifically towards species, but like as we discussed with the Crux principle, like Cynthia mentioned in her introduction, we want to study the best, or the species that's most appropriate for a question that we find particularly fascinating. So I'll let Galit add to that if she wants.
So I think the octopus is fascinating in so many ways. What reviewer doesn't want to give money for that? I mean, we are full of enthusiasm, so I think everybody should. There were quite a lot of challenges working with the octopus, because it's not traditional in many labs. There are issues with husbandry. The physiology is not entirely known, with anesthesia, a lot of protocols. There are other questions also. How do we go about neuroethics associated with octopus? So you might know that in Europe, the EU has a derivative from 2010 that regulates octopus work, similar to what they do in vertebrate, which is not the case in the United States. It is actually the case in Canada. So I think there's also a lot of questions how we go about working with these animals, which could a little slow down the activity, and also how we can use all the molecular biology and the genetic tools that are mainly targeted for mice and rodents. How can we use things like that in the mollusk? Over the last decade, there has been, I think, a big interest in using octopus. The specific type of octopus, the specific species that we are working with, which is the Octopus bimaculatus, is the only octopus that its whole genome has been sequenced. So that could be a good start to go and make a transgenic octopus, GCAMP octopus, voltage-sensitive dyes, and so genetic encoded voltage-sensitive sensors. So there are many challenges, and there's a lot to be discovered, but there's just so much to be discovered, right? So I think it's absolutely worth doing.
Right. So I think within this theme of sort of addressing challenges, I'm going to go to Paul and Angie next as representing two very different species in which you guys have successfully adapted some neurotechnology. So one of the questions we have is, what are the challenges that you've encountered with adapting some of these state-of-the-art neurotechnologies for new species, and what are some insights that you have gained through this process on how we can address these challenges? So Paul, I think.
Okay. I wanted to say that we actually, it's not right to say that we're adapting the technologies that we're using to what we're doing, because in fact, many of these technologies were developed in gastropods. So electrophysiological processes have been going on since the 1960s. So this is not new. The thing that, and what we're collaborating with Jeff Lichtman and doing the connectome, and it's actually working really, really well. And part of it is because of the similarities that we're finding with octopus, of all things. So we have to find communities where people are finding similarities, and I'm working towards doing that. Actually, Galit and I are both partnering and trying to find ways to facilitate research in mollusks, because no, it's not a mammal, but it does have advantages in many other respects. One other thing I wanted to point out with regard to the grant funding is I think it's always important to ask good questions and important questions. But when you're working on a species that does not have a large community among the reviewers, it's more incumbent upon you to even be stronger in asking the questions, the important questions, and not the little tiny bitly detailed questions that people within a community might be interested in, but not the whole neuroscience group.
Yeah, as for the bats, like from my perspective, there are like some techniques that we are now like being able to use. For example, the lab is exploring 2-photon microscopy, and some of the challenges there is that it's a brand new technique. This like has never been done in this animal, and we have to find the correct viral vectors that will express in animals that have been, for example, vaccinated against rabies. So that's a challenge that you have to think about when finding the correct viral vector. And another thing, like I'm interested particularly in molecular mechanisms and actually finding antibodies that will be good to look at things in the bat brain has been a challenge, because the genome has only recently been sequenced, and there's like a big project called the Bat1K that is sequencing the genomes of different bat species. And I think that will open a lot of doors to start using some of these molecular techniques. But it's a very, very recent development.
Corey, you wanted to add something?
Yeah, I was just going to add, I think one of the, like I always tell people in my lab, it's like a double-edged sword, right? The benefit of studying something that no one's ever studied before is that your creative capacity, like you can do anything you want, and that's really fun and exciting. The downside is that no one's ever done anything, so you're starting from the beginning, right? And so like you screw up more than you might if there were sort of tractable questions ahead of you. I mean, I think one of the things that's changed, and really, in some ways, is a little bit of a game changer, is that these sort of computational modeling approaches for tracking behavior, like people at cuts and everything else that lots of people here are using, like it's this really beautiful tool that allows you to replace years of undergraduates, like ticking boxes and so forth, right? So you can characterize behaviors a lot more quickly once you kind of train your models. And so I think this allows for, at least on the behavioral side, like a much faster curve to get at a lot of the things that you're interested in that even 5 years ago wouldn't have been possible, right? So I think hopefully that keeps people less being afraid of jumping into something new than they might have been, when really you just had to sit there for hours and hours and days and months and years watching your animals to figure out how to characterize those behaviors. So that's a real advantage that's out there for people now.
Yes, I agree. And I think also that it allows us to delve a little bit deeper, because a lot of times we had undergraduates sitting there counting, say, grid crossings or something like that. There's not much subtlety there, but there's a tremendous amount of subtlety within behavioral repertoires. So I also just kind of want to segue this conversation a little bit into genetic tools, because that seems to be a recurring theme that's come up. In particular, we have some questions for people asking about tools for transduction. And so I can speak to this briefly in the voles. So I think a general theme at this point has been that there are, at least amongst AAVs, many, many different serotypes. And one of the surprising things that we found in voles, as well as in birds, is that serotypes that don't necessarily work well in mice or rats can work very well in these other species. And so to some extent, there's an element of just test and try. It's going to become more difficult the farther away you get from mammals, because a lot of these AAVs have been designed to work in mammals. In terms of transduction for genome editing, I think that's where we start really getting into challenges related to husbandry and the bespoke nature of reproductive biology. So a good example of this is that, yes, indeed, we can make transgenic prairie voles. The first ones that we made, we used a lentivirus to transduce the genome of these organisms, which worked great for a couple of generations, but there's something about the prairie vole genome that shuts down virally inserted DNA. And so we didn't get any sort of multi-generational expression. Now we can use CRISPR to knock out different genes, but there we also had to figure out, how do we deal with a species that doesn't have an estrus cycle, that are induced ovulators? And so it's not just the technical aspect of like, what CRISPR construct should I use, but how do I effectively deploy these in these species? And so Galit, I know that you mentioned GCamp octopuses, which sounds super cool. Can you tell us a little bit about sort of what genetic tools do you see as being promising, and what challenges have you faced? Oh, and we also have a question about funding. So I can tell you for the voles, the funding actually came from NSF. But Galit, where is the funding for transgenic octopuses coming from?
So currently there are no transgenic octopus. The work that has described the sequencing of the Octopus bimaculatus genome had just came out a few years ago. So this is completely new. We don't have an octopus like that yet. We are now working on making cell cultures where we can try different viral constructs and non-viral ways to transduce the genes. We don't have it now, but this is part of our Brain Initiative work. So this is part of that mechanism. And my colleague from Dr. Clifton Rexdale, so he's a geneticist who specializes in octopus and brain development in octopus. So he's working on that aspect of the work.
Let's see. So we've discussed a bit the genetic tools and sort of some of the ways that you can approach transferring those and some of the information that you need to have available to do that. But what are some of the other neurotechnologies that your labs are currently trying to deploy within these species? And so Corey, I'll come to you first. You've been really big on behavior, and I think that that's fantastic. What are some ways that you guys are exploring some of the neurophysiology of this?
I mean, I think there are a few things, right? So a lot of it is, there are a bunch of different wireless recording technologies, right? So you can use neurologgers, you can use these full streaming wireless systems. They all have their pros and cons to this. A lot of the work is sort of trying to integrate multiple measures of behavior online with the neural recording technologies. And so this is sort of, splicing together multiple threads. And, those technologies are out there for everyone to use as long as your animal is kind of big enough to carry whatever the weight is for the system. I think the real challenge is how do you integrate the specific behavioral recordings that you're doing with that in real time in a way that is tractable, right? So that I think, for us, it's tracking gaze position and eye location, and that's sort of a non-trivial task for a monkey that's freely moving and jumping around. The neural recording part is kind of easy now. But so I think for us, it's making sure we're able to get the right things that we want out of the animal's behavior and quantifying them, and then adapting that into a behavioral recording or a neural recording system with, a closed loop system in principle to be able to also interrogate the brain simultaneously with various, optogenetic type tools. So that's sort of the next step there.
And Cindy, even though you did our opening today, I know that you actually have a long history of trying to work with some of these neurological tools in bats, even before the Brain Initiative was really sort of pushing the envelope in terms of species diversity. Can you speak to how you got that funded, and also sort of what some of the challenges you faced and any advice you would have for people in the field now?
Yeah, well, as Corey was describing the need to integrate many different measures of behavior along with the neural recordings, that's been a big challenge that we've tackled, but made some considerable progress. So we combine microphone array recordings of ultrasound, high-speed video, and in different tasks, and neural recording. So this requires an integration of the wireless recordings and time synchronized with the other measures. And originally, some of this work was actually funded by a CRCNS grant many years ago. And, we continue to seek funding for this kind of work. It's taken a lot of effort because even working with such small animals requires very lightweight devices. And because they use high frequency sounds, we have to use custom audio devices, and then the high speed video. So it's been a lot of technical challenges that now these days, I think many people are comfortable with. But when I first started many years ago with the high-speed video, we were using a camera system that was mounted to like a hot dog cart, enormous thing. And we had to use reel-to-reel tape recorders. And so now with the digital technology advances, things work much more smoothly.
Great, thanks so much. So we have a question that was posted about sort of the translational applicability of animal models, noting some of the past failures to translate. And I think part of this is an assumption that all organisms are going to be the same, despite the fact that we know from evolution that species have evolved very specific abilities in order to thrive in their specific environments, whether that's a social environment or an actual ecological environment. And so, Paul, I know that you had sort of a note about how much should we expect of translation?
I think it's important to recognize that there is no translation if you don't have basic research. You have to make the basic findings before you can translate them to humans. And nobody knows what we're going to find in basic research. If we already knew that, we would have it. So many key findings that are now commonplace. CRISPR was found because people were studying viruses and not because they were studying humans. Channel rhodopsin, again, bacterial photosensitive protein. MicroRNA was found in C. elegans. So these are features that you need the basic research for before you can even think of applying them to humans. So it's not a matter of one taking away from the other, it's that these work together.
All right, and so we're gonna wrap up here, and what I'm gonna do is ask everybody just for a quick one line, what is your favorite thing that we've learned on a basic science perspective from your species or multiple species of interest? And so I'll start off. Paul mentioned this in his talk, but I am absolutely enamored with how much we've learned about the highly conserved role of social neuropeptides, specifically oxytocin and vasopressin, from studying it at a very basic level and now beginning to understand how it may affect human social behavior. So Corey, you're next.
I guess, a lot of the things that I think I've been most excited about that we've learned with the marmosets is that a lot of the sort of assumptions about how the primate brain work using more traditional approaches haven't really panned out to be predictive of what the brain does in the natural states as the animals are really navigating social circumstances or really almost anything we've tried. So I'm excited to see as we continue kind of pulling those threads, how our view of how the primate brain really works changes as we go.
Great, Andy?
Yeah, well, there's so many things I can say about the wonders of bats and what we've learned from them. I guess one thing that I am very, very interested in is how they are able to discriminate sounds that are overlapping and really making sense of these cocktail parties. And we've learned a lot about the auditory system in studying this, but also things like 3D spatial navigation and 3D scene representation, which are really novel because you can't really study this in other species. If you think about place cells in mice and rats, it's all in a two-dimensional plane, but when you study it in bats, now you're suddenly in a three-dimensional space, which is really, really interesting.
Thank you. Andres, you're up next.
Sure. Maybe I shouldn't find this surprising, but I still do every time I work with animals in the lab, is how they can maintain so much of their natural behaviors of even in the control laboratory conditions we keep them in. We try to make it as ethologically relevant as possible, but that they will still fight or that they will communicate, that they do so many of their things in these very pristine control conditions that it works to an advantage for us that we can study them carefully. But I find it surprising how much they still behave naturally, let's say.
Great. And Paul, you're up next.
I think it's really important and striking that you have to understand the animal's Umwelt. This is the ethological phrase about the environment that the animal experiences, not the environment that we think that they're experiencing, because they're not us. They are different. And we can learn from those differences. And that's what I think is most striking.
Perfect. I think that Paul put it much better than I did.
All right, and Galit, you want to round us out?
Just a couple of specific things about octopuses. The brain is somewhat different than ours in its organization. It doesn't have the somatotopic organization the mammalian brain has. And yet, the octopus in my lab, they enjoy playing with Legos. They seek they are so playful and they are so creative and they initiate play. So maybe what we consider in our brains, right, how things work are not the only way that they can work. And their evolution came with so many creative ways to make just amazing behaviors. And our octopus has a favorite arm to use, even though all the arms are exactly the same. So their biology is just amazing. And we have to keep exploring.
That's fantastic. And Cindy, can you sort of sum it all up for us with one final thought? And I think then we'll go ahead and close. You're muted, Cindy.
Sorry about that. We've heard some fascinating examples of animal that can really provide important insights to neuroscience, that they can really answer some of the bigger questions, and we need to forge ahead and continue with this line of comparative research.
Thank you so much. So with that, I'd like to thank our speakers as well as NINDS, the Brain Initiative, and also everybody who tuned in today. Thank you for your attention.